What if you could change the genetic makeup of a cell? Would you press select, cut away all the parts from the genetic code that you just don't want anymore? Now, how about if you could do that for an entire organism? Would you do it then? Biologists have this opportunity every single day using CRISPR-Cas9 gene editing tools. These tools completely revolutionize the world of gene editing. And essentially, it's just like taking a pair of scissors and cutting away from a piece of paper the stuff you just don't want anymore. You can do that with DNA. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which I know sounds like a mouthful, but is actually the name of a series of nucleic acid sequences found in prokaryotic cells. The nucleic acid sequences in these prokaryotic cells were found to have copies of virus genes that were stolen out of virus cells themselves. Now, before I go more into what CRISPR is, let me just explain a little bit behind the biology terms involved. Genes are composed in sequences of 3. Each of these sequences is extremely specific, and changing just one of them can result in changing the functionality of the entire cell. For example, if we have the genetic sequence CTC, we have a cytosine, a thymine, and a cytosine. Changing just one of these from thymine to adenine results in the amino acid being coded for changing from valine to glutamic acid. This causes sickle cell anemia, which is an extremely harmful disease caused by a genetic mutation just like this. Now, CRISPR technology uses short single-guide RNA, also known as sgRNA, to guide the Cas9 enzyme to the target site within DNA. Once its target site is reached, it initiates a double-strand break of the DNA, which then in turn also initiates either DNA repair pathways or DNA deletion pathways based on what we want to do with that particular gene, mostly based on the mutation that is found in that particular gene. Essentially, biologists have the ability to take a pair of scissors, cut into the genetic code of an organism, pull out part of the genetic code, or replace it just as easily again. This technology was developed by 2 leading women in science, Dr. Emmanuelle Charpentier and Dr. Jennifer Doudna. They first published their findings on CRISPR gene editing in 2012 and have since completely revolutionized the biological world and gene editing processes. CRISPR technology has been used to improve medicines such as antibiotics and to cure diseases such as hereditary blindness. In November of 2020, these leading women in science won the Nobel Prize in Chemistry for their creation. They became the first women to win a Nobel Prize in any scientific field without the assistance of a man. They continue to revolutionize the scientific world while breaking down barriers for women in STEM every single day.